Bismillahirrahmanirrahim. Dear students, I hope you all are doing great and I welcome you all in second year Pharm.D. This is our very first lecture of Pharmacology lab, code Pharm-314, session morning and evening on the study of basics of pharmacological experiments and the introduction of experimental pharmacology. So before I begin, I would like to inform you all that the manual of this lab and the reading material shall be uploaded on the Google class whose the code was previously shared. The objectives of the introduction in pharmacological experiments are: after this presentation, you all shall be able to define what are the general considerations required in pharmacological experiments. What is the rationale and reasoning behind pharmacological experiments? What are the types of experimental testing? You should also be able to understand what is a clinical trial, what is a preclinical testing or study, what are the types of technique used in preclinical testing, what are the different types of animals used in lab and research, and the regulations and ethics and laws in animal testing required in experimental pharmacology. So, to understand the basic concept and various terms and approaches used in experimental pharmacology, we should have some background information. The drugs and their use were well started in prehistoric era. Even the beneficial and toxic effects of many plants, animal resources were recognized. The concept of pharmacology began in 17th century when the observation and experimentation began to replace traditional drug use. Many historians and physicians started to understand the value of experimentation when they performed experiments to evaluate effects of some traditional drugs used in their own practices in the treatment of several diseases. Thus, the science of drug preparation and therapeutic use of drugs began to develop as the part of pharmacology. But the limitation was the lack of facilities regarding methods for obtaining active constituents from different sources. For that, we should know what is pharmacology. To overview, pharmacology is the branch of science which deals with study of drugs on the living system. Now, what is experimental pharmacology? It deals with the study of effect of various pharmacological agents on different animal species. What is the purpose of pharmacological experiment? The purpose of pharmacological experiments are to discover new therapeutic agent, to discover new use of existing therapeutic agent, to study the toxicity of drugs, and to study the mechanism of action and the site of action of drugs. As the pharmacology is the study of drug, so a quick definition what a drug is. According to the WHO, a drug is any substance or a product that is used for the modification of physiological system or pathological state, means any disease state for the benefit of the recipient. It is also defined as any chemical substance which is used for prevention, treatment, or the diagnosis of a disease. Moreover, if I ask you about the reasoning or the rationale behind doing this experiment in laboratory, then what is the purpose of only doing experiments in pharmacology? It basically helps in better understanding of the theory, what you will learn in the class. However, also to appreciate such principle and the importance of testing drugs for their effects. Practical classes also aim to illustrate such effects on isolated and whole animal preparations. Moreover, you will also learn the different handling techniques of animal used in pharmacology lab and the pharmacokinetic parameters, which is the movement of the drug in and out of the body and what the body does with the drug. Also the pharmacodynamic principles, which is the mechanism of action and the biological response of the body towards the drug. It also helps in complementing the principles of physiological systems you have learned in first year. Experimental pharmacology comes under the heading clinical testing, preclinical testing, which are subdivided into in vivo, ex vivo, and in vitro testing. And there are some approaches in pharmacological experiments, which I will discuss later. Before that, I would like to tell you about the pharmacological evaluation of drug. Just a brief overview of this. As you know, study of drug is pharmacology, and there are sub branches of this, which are pharmacokinetic and pharmacodynamic. Pharmacokinetic is what does the body do to the drug, and it is the movement of the drug throughout your body. So when you ingest any tablet, where it goes and how it absorb and distribute and metabolize and eliminate into the body, this is all in pharmacokinetic. Then comes the pharmacodynamic. This is what does the drug do to the body. It is the mechanism of action, you can say that, and the biological response of the body towards the drug. So when the drug reaches to the systemic circulation, where it goes to the target site and what receptor it binds with and which inhibits, if it can be agonist or antagonist, we will discuss that later. So pharmacodynamic is that. Evaluation. What is evaluation? It is the examination based on various criteria, especially safety and efficacy. So safety and efficacy are very much important for any drug. Obviously, every drug has some unwanted effects besides its indications, besides its uses. Drug development process comes under pharmacological evaluation of the drug. So it is a process of bringing a novel drug from bench to bedside. It almost can take 10 to 15 years for a drug to be designed, developed, and approved for the use in patients. Before a drug can reach a patient, it must go through rigorous testing to determine whether it is safe, effective at treating the condition it was developed for, and to ascertain the correct dosage and appropriate administration route. There are several pharmaceutical regulatory authorities which are responsible for the supervising and regulating therapeutics, including prescription, over-the-counter drug and vaccine. There are various stages of developing a drug. Number one is the basic research, which includes thousands of compounds. From that thousands of compounds, we screen these compounds into 5,000 to 20,000. That comes under drug discovery. In drug discovery, the target identification and validation, hit identification and validation, and hit to lead optimization is done. And it from the lead optimization, it takes to candidate selection. What is a candidate selection? At this stage, we determine from the several promising high quality leads. What is a lead? Lead is a target compound, from a novel target compound from thousands and millions of compounds. We screen to 20,000 and then we come to 250 compounds only in preclinical testing, that is animal testing or in vitro or in silico testing. For pharmacodynamic and kinetic parameters we do for the pharmacological evaluation, and from that We screen to 5 in clinical research, that is on human, and then comes to the one that finally is the approved drug. Tutorial on the drug development process presented by Novartis Pharmaceutical Industry. You may also want to have a look on this.
In drug research and development, the patient is the main focus. The mission for Novartis is to help the patient overcome a disease and improve their quality of life. The drug development process is designed to ensure that innovative new medicines are effective, safe, and available for patients in the shortest possible time. The first step in drug development is to discover the best targets for treating or preventing a disease. Targets are usually proteins in the patient's body, which are associated with a disease, or proteins in microorganisms causing a disease. The challenge is to identify which proteins are relevant and, more importantly, confirm their role in a disease. Increasingly, Novartis focuses on understanding cellular networks of proteins, or pathways. A single protein may transmit messages to several other proteins, sometimes in multiple pathways, affecting their function. Knowing how these pathways work and interact helps to identify the most appropriate target for a drug. The pathway approach allows Novartis to better understand the mechanisms of a disease. This knowledge, together with the desire to address unmet medical needs, determines the priorities in target discovery. In drug discovery, several methods like high-throughput screening and computer-based design are used to find chemical compounds or biologics that bind to the identified target. If a compound modulates the target in a way that is expected to alter the disease, this so-called hit will be refined to improve its safety and effectiveness, eventually becoming a drug candidate. Discovering and bringing one new drug to the market typically takes an average of 14 years of research and clinical development efforts and costs around $2 billion. Of 10,000 or more hits tested in early drug discovery, only one may eventually lead to a drug that reaches the market. In the late preclinical stage, further experiments are conducted on the drug candidate to ensure it is safe for patients and has the required pharmacokinetic properties, like appropriate absorption and metabolism by the human body. These experiments are executed with extraordinary diligence to minimize any risks to human test subjects. Animals play a critical role in the drug discovery process as well. Although much research and development can be done using various experiments or using computers, complex disease mechanisms can often only be understood through the use of animal studies. Also, governments and regulatory authorities require that medicines be tested in animals before they are tested in humans. Novartis keeps this research as limited as possible and always ensures that animal research is scientifically acceptable according to current standards and regulations. For these reasons, Novartis continues to use animals in its quest to find innovative, safe, and life-saving medicines for patients. At the same time, Novartis is committed to refining, reducing, and replacing the use of animals in research and upholding the highest standards in animal welfare.
So this
Programs consist of several phases, each of which is focused on evaluating drug safety and effectiveness or efficacy. Information on ongoing phases. Clinical trial programs consist of several phases, each of which is focused on evaluating drug safety and effectiveness or efficacy. Information on ongoing clinical trials can be found at www.clinicaltrials.gov. In Phase 1 of clinical trials, the drug is usually tested in healthy volunteers to determine its safety and pharmacokinetics. In Phase 2, the drug is given to a group of about 100 to 250 patients with the disease to evaluate its efficacy and to determine the optimal dose. In addition, the safety of the drug and its side effects are evaluated, as these may be different in patients compared to the healthy volunteers tested in Phase 1. Novartis also conducts trials called Proof of Concept or POC trials, which are often a mix of Phase 1 and Phase 2 studies. A group of well-defined patients is treated with the drug candidate to provide an early indication whether it actually affects the target in the intended way or has a beneficial impact on the disease. These studies allow Novartis to rapidly invest resources on drug compounds, which are effective and safe. This way, the appropriate medicines can reach the patients faster. The data from Phase 1 and Phase 2 studies provide the scientific confidence necessary to continue the development of the compound in larger clinical trials. In Phase 3 of clinical trials, 1,000 to 3,000 or even more patients are recruited for research with the investigational drug. Phase 3 clinical trials are undertaken to confirm the effectiveness of the new drug, monitor side effects, compare it to established treatments, and gather additional information to allow the drug to be used safely. To bring a new medicine on the market, every new drug needs to be registered by the health authority. Data from all clinical trials are collected and compiled into a document called Registration Dossier. The Registration Dossier will include efficacy and safety data to support the final intended use of the drug. The dossier is then customized for different regions and countries around the world.
Student, this dossier means that it contains all the information related with the clinical and preclinical testing about the compound or the drug. So in Pakistan, we have Drug Regulatory Authority of Pakistan, which accepts these kind of dossiers, and pharmaceutical companies, they submit this dossier to the DRAP for the acceptance of the drug or compound or generic they want to have for their company.
To meet the requirements of health authorities. This is a crucial step to ensure that patients all over the globe the requirements of health authorities. This is a crucial step to ensure that patients all over the globe can access the innovative medicines that have made it through the complex drug discovery and development process. Once the registration dossier is approved and the product is launched, several activities will continue, like drug safety monitoring, which is mandatory. Post-authorization safety updates, annual reports, and any additional information required by the health authority must be provided at defined intervals as long as the drug remains on the market. Often, Phase 4 clinical trials are initiated to gather additional data not collected in the Phase 3 program. This may include efficacy, safety, additional benefit and risk information, as well as pharmacoeconomic data. Our mission is to provide safe and effective treatments that benefit patients and improve the quality of their lives.
Students, many questions can be extracted from this video. From the portion of drug discovery, the question can be: what is HIT, what is lead optimization, what is a candidate selection. I may ask, from the preclinical testing, the types of preclinical testing like in vitro, in vivo, in silico testing, and why we use animal in preclinical testing, what is the purpose of preclinical testing. For example, it is used for the efficacy and safety, and then comes the clinical trial. What are the phases of clinical trial? There are four phases of clinical trial: phase 1, phase 2, phase 3. Phase 1 includes the healthy volunteers, phase 2 includes the healthy volunteers as well as the disease patient, and third phase include more number of population, and phase 4 is post marketing surveillance and pharmacovigilance. So these questions can be asked from this video. You should watch this over and over. Students, you already would have got enough idea about the pharmacological testing, animal testing, which are done for the pharmacological studies and toxicological studies. For toxicology, we assess its safety, and for pharmacological study, we assess its efficacy. And then after that, the animal to human dose is converted, and then the compound is sent for the US FDA approval for the clinical trials. This is for pharmacological testing can be: number one is the use of ancient or folk remedies, herbal preparation for the determination of their possible pharmacological activity. And the folk remedies are the traditional herbal medicinal plant. For example, salicylic acid is well known for its potent anti-inflammatory effects. Um, it was used by the Native Americans, and they used to brew it and used as a tea for the ailments like inflammation. Another example is the evaluation of anti-inflammatory effect of turmeric, curcuma longa, on the basis of its use in regional tribes of India for pain conditions. So turmeric is haldi and its scientific name is curcuma longa. It is widely used everywhere and it is derived from India for pain. Another example I would like to tell is the use of the cinchona bark. Cinchona bark was used by the South American to treat the chills and fever. Further, it was researched, and it was researched which led to the development of compound quinidine and quinine. Subsequently, it was used to treat malaria and cardiac arrhythmias respectively. Another approach is the screening of an old existing drug for their new possible therapeutic effect. These are the already marketed drugs, and if we want to identify their new indication or use. Examples are atorvastatin. Atorvastatin is a cholesterol lowering drug found to be effective in reducing symptoms of multiple sclerosis. It is a disease in mice, leads to the development of drug with new pharmacological activity. So let me write it over here. Where is third approach is the structural optimization or the modification of existing drug for their modified effect. It means the modification of the structure of any compound to increase or improve its efficacy, safety, and therapeutic effect, and the determination of pharmacophores. So the structural optimization of lead compound, for example, salicylic acid by the company Bayer, resulted in the development or the discovery of acetyl salicylic acid, well known and popularly known as aspirin. It is the first non-steroidal anti-inflammatory agent. And so the third approach is structural optimization or modification in existing drugs for their modified effects in terms of improved efficacy, safety, or new therapeutic target, and the determination of pharmacophore. It means the modification of the structure of any compound which leads to the development or the discovery of a new compound or the new drug. For example, a structural optimization of a target compound, which is a salicylic acid derived from the willow bark by the company Bayer, resulted in the development and discovery of acetyl salicylic acid, popularly known as aspirin. It is the first non-steroidal anti-inflammatory agent. Another example of structural optimization is the potassium channel agonist dioxazide, which was discovered after the thiazide diuretic such as hydrochlorothiazide. It exhibited diuretic activity. It causes diuresis activity. So the structural modification of this diuretic compound lead to the development of diazoxide. Diazoxide is a vasodilator. Both drugs can be given for hypertension. The general consideration for pharmacological experiments. For any preclinical pharmacological experimentation, following points must be considered. Number 1 is animal selection. Selection of experimental animal is made in accordance with the objective of the study. Different animal-related factors like age, weight, sex, and species may affect the result and should be considered during selection. Number 2 is dose calculation. Dose for the experimental subject is appropriately calculated according to the weight of subject and test dose of new drugs and established human dose of old drugs. Third is selection of vehicle or physiological solution. Most commonly used vehicle for making dilution of different drugs and physiological solution is normal saline. So, in short, vehicle is the like normal saline which dilutes the drug or the compound. The selection of the appropriate vehicle is important according to the solubility nature of test drug. If the test drug is not soluble, then there is no reason to conduct the study further. And the route of administration. It also depends on the route of administration as well. Other common vehicle used include number 1. The most commonly used vehicles are polysorbate 80, which is the generic name is 2-in-80, and it is a non-ionic surfactant and emulsifier. This is a synthetic compound and is viscous, water soluble, yellow in liquid. It is frequently used to increase solubility of test drug in water. Another is saline, which is a normal saline. It is isotonic solution of sodium chloride 0.9% weight by volume. Next is CMC, DMSO. What is DMSO? Dimethyl sulfoxide. It is a compound and it is colorless liquid. It is an important polaroprotic solvent that dissolve both polar and non-polar. So, it is a compound which dissolve both polar and non-polar compound, and it is miscible in many organic solvent, for example water. CMC is carboxymethyl cellulose, also known as cellulose gum, derived It is a derivative actually of carboxymethyl groups. It is often used as sodium salt, sodium carboxymethyl cellulose. The grouping of animals. Animals should be divided into two major groups before starting of any pharmacological activity. That is the test group. It is the group of animal in which test dose of the test drug is given. Test drug is the drug which we want to test. The control group is the group of animal in which simple saline or any other vehicle is given and that serve as the control for measurement of pharmacological activity of test compound. For both of the group, all the parameter related to animal housing and food should be the same. Next, most important consideration is the development of experimental model. Excuse me. Different models are required for the experimental purpose according to the objective of study. For example, aged rodents. Rodents, what are rodents? Rodents are rats, mice, the animals which have the front teeth which are sharp, very sharp. For example, rabbits, for example, rats, mice, guinea pig, hamster, they all are rodents. So, aged rodents and scopolamine induced amnesia. Scopolamine induced amnesia for Alzheimer's. Scopolamine is a muscarinic acetylcholine receptor antagonist which disturbs learning and memory, and it causes short-term amnesia. Amnesia is the loss of memory, and it is a useful model for the disease Alzheimer's. Another model is very most popular model, that is formalin model. So the rodent formalin model is used as an acute and rapid in vivo screening study for the evaluation of potential analgesic effects of novel chemical entities. What is an analgesic drug? The drug which reduces the pain. So the injection of formalin into the rodent, mice or rat, hind paw induces biphasic nociceptive behavioral responses. What is nociceptive? Nociceptive is related with the pain. It produces pain. So this was all about the introduction and basics of experimental pharmacology, and do watch it over and over. If anything you did not understand, you can ask anytime. Thank you so much.